Good morning or afternoon, everyone. Welcome to today's webinar. My name is Marie Latulippe, and I am the Director of Science Programs at the Institute for the Advancement of Food and Nutrition Sciences, or IFNS. And for those of you who might be joining us for the first time, we wanted to let you know a little bit about our organization. So IFNS connects our tripartite membership, so scientists from government, industry, and academia, to support collaboration and catalyze knowledge in the areas of advancing food and nutrition science for public benefit. And one thing we do is that we actually fund actionable research that is conducted by external researchers that are selected through an open competitive process. And all of our projects are actually viewable on our website if you want to take a look at what we are funding. We advance food and nutrition science for public benefit by convening experts across sectors to engage in dialogue. And we do that by combining funding from the public and the private sectors, as well as in-kind contributions. And this slide is showing the overall mission and focus areas of the committee that's bringing you this webinar today. And this is the third in our webinar series that's focused on the role of diet and gut microbiome in immune response and inflammation. And our intention here is to explore these relationships from a variety of perspectives to understand the state of the science and possibly identify gaps that could be addressed through IFNS programs. And a quick note on logistics for the webinar. What we will do is we'll go through the presentation and then take questions at the end. So we do encourage everyone to type your questions in that questions box as we go along, and then we'll bring them to the presenter at the close of the presentation. So with that, we are extremely pleased to introduce to you Dr. Paul O'Toole, who is a leader in this field. He is a professor of microbial genomics and the head of microbiology at University College Cork in Ireland. He's also a principal investigator in APC Microbiome Ireland, in which he leads projects on gut microbiota as a modulator of health and disease risk. And his main research program is on the functional genomics of GI bacteria in humans, with an emphasis on gut microbiota in aging, colorectal cancer, and IBS. And the ultimate aim of his work is to understand and control host microbiome interactions, especially in the context of multifactorial disease. So Professor O'Toole, if you'd like to join us by webcam, he will speak for about 30-35 minutes, and then we'll open for audience questions. And at the end, we will be closing so that members of the IFNS Gut Microbiome Committee can engage in a dialogue. So I will turn the presenter rights over to you, Dr. O'Toole.
Okay, thanks, Marie. Can you see my title slide?
Yep, looks perfect.
Excellent. Okay, good morning or good afternoon, everybody, wherever you are. It's 3:05 here on the southwest coast of Ireland. It's a lovely sunny day, and I'm delighted to be here. I'm going to share with you our recent research on the role of the microbiome in healthy aging, with a focus on, as Marie said, inflammaging. And I'm going to try and make it relatively light on the microbiology. Full disclosure, I'm not a nutritionist. I know nothing about nutrition. Well, that's not quite true. I've picked up a little bit for the last 5 years, but I'm primarily a microbiologist. And when I talk to people not fortunate enough to be microbiologists, I explain that the gut microbiome is the variable community of microbes in your gut, although the microbiome can be anywhere, and it includes a lot of different small living things, such as the bacteria, archaea, etc., which are listed on this slide. And some of these organisms are living there full time, they're residents. Some of them are tourists just passing through. Some of them may be invited guests, such as probiotics, or others may be vandals, such as enteric pathogens. And the microbiome exploded as a field of research and endeavor in the last 20 years because it became recognized that we harbored this really complex community of organisms which have co-evolved with humans and which perform a large number of necessary functions. And there are some baffling statistics which are listed on this slide, which will be available upon request to Marie as a PDF. We became interested not because we like numbers, but because the microbiome is intrinsically linked to human health. It's malleable. In other words, you can change it. You can change your genome, and nutrigenomics is a fascinating field in its own right. But if you've got dud genes, there's nothing you can do about it. That's what you got, whereas your microbiome is changeable. And some of the things going on in your life which change your microbiome are age, diet, the kind of person you are, the kind of animal, or antibiotics. And these factors influence the microbiota in different kinds of ways. And where we're kind of going with this research is that we want to control the microbiota in a precise way to promote health. And obviously, I'm going to talk to you about that in the context of healthy aging. The microbiome has been proposed as a contributor to disease for lots of different diseases, and examples are inflammatory bowel disease, where you've got an altered microbiome which gets close to the intestinal epithelium. Irritable bowel syndrome, where the major effect is probably a metabolic one, where excess fermentation creates acidic end products and gas, and that exacerbates people with visceral hypersensitivity and triggers the gut-brain axis. Microbes even contribute to the risk factors for atherosclerosis because they convert macrophages into foam cells, and they do this by generating a metabolite called trimethylamine oxide, or TMAO. And I mentioned that in passing because I'll come back to it later in my talk. And basically this metabolite plus these transformed macrophages blocks your arteries and forms a plaque which can come off and give you a stroke. And lastly, and more sinisterly, microbes appear to contribute to the evolution of colon cancer, and we have a program, as Marie mentioned, in my lab working on that. So the general point I'm trying to make here is that the microbiome is a risk factor for a number of diseases. And I teach genetics for a living, and I tell the students the microbiome doesn't cause any of these diseases because most of them are multifactorial anyway. And the microbiome is at best a contributory environmental factor, but it's one which we have reasonable chance of being able to manipulate. In terms of what your microbiome looks like, you're born essentially devoid of microbes, and throughout the course of the first couple of months and years of life, you assemble a complex microbiome through a series of chaotic successions and replacements till about age 1.5 or 2, when you probably have an adult-like microbiome. And that's stable throughout adulthood until you come to old age, which is a movable threshold, and then the microbiome goes into a state of flux again. And it had already been shown using plate and loop microbiology 20, 30 years ago or longer that the microbiome was changing in older people. So when we commenced this research, the reasons for doing so were the following. The proportion of people over age 65 is increasing in the world, which is a good thing. And this is a testament to modern medicine. But these people, when they constitute a larger proportion of society, have larger healthcare costs, and they have a justifiable expectation of enjoying their golden years and enjoying health span, not just lifespan. As I said a moment ago, it was already known that there were changes in the microbiota composition and activity, and it was suggested but not proven that they might contribute to different rates of loss of health as a function of aging. What's always been known but tends to be overlooked is that in older people there are increased infection rates. 30% of the hospitalizations of people over age 65 are recurrent urinary tract infections, which, if they don't have a gut source, certainly have a gut microbiome link. There are also increased incidences and prevalences of inflammatory diseases. And because inflammation is one of the major themes of this talk, let me just define what I mean by that in case this is not your field. Inflammation does not mean when you hit your thumb with a hammer and it becomes inflamed. Inflammation in this context means persistent and unwanted activity of the innate immune system, and that can be measured by looking at the activity of innate immune cells, or it can be simply measured in a person in your office by looking for inflammatory chemokines or cytokines, or measures like IL-6 or CRP. And this persistent inflammatory state means that the innate immune system is chasing shadows instead of reacting to pneumovaccines or COVID vaccines or winter colds. So all of this added up to us to the prospects of dietary intervention or modulation if we could show that the diet was modulating the microbiome. And this is a cartoon reminding you that there are a number of different factors going on in aging, including epigenetics, genetics, stochastic, random effects, and also the environment. So I'm including the microbiome as an environmental mediator of disease risk. And my colleague and friend Claudio Franceschi in Bologna coined this term inflammaging, which is the persistent activity of the innate immune system in older people. And that's got interactions with a whole raft of body systems, including the gut immune system in general, also the metabolic system, which is not well represented on this slide. When we started working on this, the first thing that we did is we established a national cohort called ElderMet, and we profiled the microbiome using sequence-based methods in a series of papers. And in these representations, each of the colored circles is a person, and the position of that person relative to all the others is determined by the relatedness of their microbiome. So if two people have very similar microbiomes, they're very close, and if two people are very far apart, they have very dissimilar microbiomes. And the only other detail is the color scheme, which indicates where the people live, either in the community, buying their own food and preparing their own meals, or in long-term residential care, what we call long stay. So it appears that where you live in the community in Southern Ireland determines the kind of microbiome you have. And if I circle the major foci of those individuals, that becomes a lot clearer. Now, we wondered what was driving this separation, and to make a long story short, we used a number of statistical methods. This is one called orthogonal least squares. And now for each person, we have a line. And at one end of the line, we have the dietary data, habitual dietary data, represented by a validated food frequency questionnaire. And the other end of the line for that person is their microbiome data. If there was no relationship between diet and microbiome, these lines would be all over the place, crisscrossing in a tangle, which is clearly not the case. There's a relationship between the habitual diet and the microbiome. And in fact, in follow-up studies, we see that when people move from the community into long-term residential care, their diet changes immediately, but it takes a while for their microbiome to change. Now, because this is a nutrition type audience, you may be asking, well, what are these diets? And broadly speaking, we can define them into four major dietary groups, where the best diet is dietary group 1, and the worst dietary group is dietary group 4, ranging from low fat, high fiber to high fat and moderate fiber. And while we were doing this research, other scientists in North America, particularly Gary Wu in Philadelphia, were looking at the effect of these kinds of diets on the microbiome. The health indices in this discovery cohort ElderMet were linked to differences in the microbiome. And I appreciate this is a complex table, but I have helpfully arrowed all of the phenotypic markers which statistically significantly associate with the microbiome. And they include the geriatric depression test, a nutrition assay, blood pressure, several indices of frailty, calf circumference, and 2 markers for inflammation, IL-8 and CRP. So in our first paper 11 years ago now, we saw statistically robust links between the microbiome and these health indices. Okay, so, and we work closely with gerontologists and clinician scientists, and they reassured us that the delta values across these cohorts were not just statistically significant, they were clinically significant. In other words, the difference in CRP and IL-6 levels between the people with the poorest microbiomes and the best microbiomes corresponded to clinically significant differences. The inherent weakness in this kind of study was the fact that we went out and recruited people living in the community who were healthy and people in long-term residential care who were frail. They were in care for a reason, they needed help. So we went back to the ElderMed expanded cohort and we focused on people who were community dwellers, but who voluntarily or unwittingly consumed a long stay-like diet. So these are three groups of consumers. We've banded them into three diet types. These are people with a healthy diet. This is the color scheme. And you can simplify it by saying that stuff that's in green is good, like low-fat dairy or vegetables or fruit and fiber, and stuff in red and yellow is bad. So these are people living in the community eating a prudent diet. Then there's a second group that consume a less healthy diet, and then there are people living in the community consuming what we call a long stay-like diet for various reasons, which could be sad social reasons, family circumstances, or maybe just lack of education. Anyway, they consume a diet like people in long-term residential care. And importantly, these individuals with the long stay-like diet have a long stay-like microbiome. In other words, it's not necessary to be in long-term residential care to consume this low complexity diet, high fat, low fiber, and your microbiome is altered as a consequence. And these are 4 clinical measures, and the long stay-like group corresponds to the last 2 microbiome-based divisions. So this is the third long stay-like category in the previous slide, and they have lower measures of functional independence. In other words, these individuals are frailer. They have measurably and clinically significantly lower levels of cognitive function as measured by the mini mental state exam. They're more inflamed, and there's a trend towards lower calf circumference, meaning potentially onset of sarcopenia. So we're relieved to see this because it validated the hypothesis that the microbiome was at least correlated with inferior health scores. We've spent a lot of time trying to distinguish between microbiome associations with age and microbiome associations with other diseases because, well, not that age is a disease. A number of conditions, including IBD, type 2 diabetes, cirrhosis, colon cancer, and polyps, are characterized by microbiome alterations. And we're starting to realize that many of these alterations are the same across multiple diseases and may be consequential to the disease. So we have developed age-aware signatures for diseases. In other words, you can detect IBD microbiome associations better if you know the age of the patient, and you can distinguish between microbiome signatures for these diseases and with aging. So that's just a finesse, but I think it's an important one. So to summarize this opening part of the talk, the gut microbiome of older people interacts with age, with diet and health. The directionality and the health effect is implied rather than proven. The habitual diet impacts on diet and health independent of where you're living. And there are low overlapping but distinct microbiome signatures for aging and dysbiosis-related disease. Now, we're painfully aware of the fact that correlation is not causation. And obviously, we wanted to unravel the directionality. In other words, are these microbiome differences consequential of aging and poor health, or are they contributory or even symptom exacerbating? So we designed a couple of trials, which I'll go quickly through. The first one was we colonized germ-free mice with the microbiome from healthy seniors or frail older people, and we put them on different diets corresponding to community or long stay, and we phenotyped them. And then we did a 6-month dietary fiber supplementation aiming to try and improve the diet of people living in long-term residential care. Now, either of these experiments could constitute a seminar in their own right. So I'm going to take the higher road on this and just stick to very simplistic summaries. This is the experiment where we colonized mice. This is the original murine microbiota because these were conventional mice that we treated with antibiotics. This is the microbiota of the healthy donor, and this is the microbiota of the recipient mice on the first 4 weeks when they were receiving, I'll call, a good diet. And then we transfer them to a high-fat, low-fiber diet corresponding to that typical for long-stay residential care consumers. And all you have to do is to look at the blowout in the red color. It's quite easy to promote the outgrowth of bacteria, which we know to be associated with unhealthy aging in a colonized mouse simply by feeding them a low-quality diet. The corollary experiment is to colonize the mice with the long-stay microbiota. Now you can see there are more red parts of the pie to begin with and less blue. And then we transfer them to a low-fat, high-fiber diet. And you don't see the reciprocal switch, or as I've said in previous talks, which tends to be the sound bite that people remember, it's easier to make a good microbiome bad by a diet switch than it is to make a bad microbiome good on a diet switch, simply because some of these taxa seem, well, at least in mice, do not seem to respond well to these dietary switches. And we did some immune profiling, which I'm going to skip for the reasons of time, and they were largely non-significant anyway. The more exciting experiment was obviously to do in humans, a lot trickier. So we took people who were living in long-term residential care, and we gave them a 6-month prebiotic supplementation. There was actually a 2-week run-in, and then a 6-month feeding, and then a 6-week washout. And we profiled the microbiome at all those marked points by ribosomal RNA gene sequencing. And just to make it challenging, we also did a prebiotic supplementation in healthy seniors living in the community and in young healthies, which we scared up around our university. Briefly on the prebiotic intervention, I should have mentioned this trial was done in collaboration with General Mills for full disclosure, but they didn't restrict our choice of prebiotics at all. They just wanted to sponsor some discovery science. We aimed to give these folks an additional 21 grams of fiber a day, which was a significant increase for these kinds of people. And we made a mix comprising ingredients from the literature, and you can see the references here, and we tried to choose ingredients that had been proven to improve the microbiome in humans rather than in experimental animals. Were we to do this again, in hindsight, I probably wouldn't choose so many ingredients, and I probably wouldn't choose the same ones, but this seemed like a chance to give us an effect at the time. Now, for the next couple of minutes, I'm going to show you pairs of panels where we have the subject type on the intervention or placebo as a function of the trial period. So these are the young, healthy people who got the intervention or the placebo for the 6 months plus washout. Then you've got the elderly community intervention placebo, and then the more interesting group, the elderly long-stay intervention placebo. And what we're actually looking at here on the y-axis are the observed species. This is a very crude summary index of how good your microbiome is. And if you've been looking at this literature, you'll see measures of so-called alpha diversity. And this corresponds to how many species, or sometimes it's listed as a Shannon index or Simpson index. And you can see that the number of species in the long-stay individuals is lower than the other two groups to begin with, which is what you would expect. But the important outcome is that the prebiotic intervention did not alter or improve the microbiota diversity after 26 weeks. There were some altered abundances of some taxa. The differences were significant for a while in the long-stay intervention group, but none of this was enough to say that it was spectacularly successful, and nor were the alterations in chemokines or inflammatory markers such as IL-6, CXCL11, or increased CD4 plus T cells, which is one of the things you want to get, because these are things that older people with tired immune systems tend to lack, that they lose naive CD4 plus T cells. So we were kind of dispirited at this point because it was an expensive experiment. It involved both an animal trial and a human trial, a 6-month intervention, 20 grams of prebiotics. It was a pretty ambitious experiment, and the outcomes were relatively modest. Now, other people have reported reasonably successful fiber interventions. At least they tend to increase the proportion of lactobacilli and bifidobacteria, which doesn't excite me too much. if you look at the broader scientific literature, we know that animals that consume different different diets have different microbiomes. So this is work published years ago by Jeff Gordon's group, I think. And basically the color code tells you that herbivores have a very different microbiome from omnivores or strict carnivores. So I was lucky enough to be part of a joint programming initiative project with a guy in Naples called Danilo Ercolini. And he had access to a really nice cohort of omnivores, vegans, and vegetarians in Naples. And this heat map shows that the 3 dietary consumption pattern types have different microbiotas. So this is a heat map where red means a lot or high correlation. So you can see that the omnivores, these food ingredients up the top, are meats and dairy and stuff. So the omnivores have loads of these bacteria and very little of these. In the vegans and the vegetarians have a little bit more, whereas down at the other end of the chart with the plant-related food ingredients, the vegans have loads of these bacteria, lactospiraceae and Prevotella. The vegans have some but not as much, and the omnivores are relatively little. So why am I telling you this? I'm telling you this because one of the metabolites that we could readily measure was the one I mentioned earlier on, TMAO. And you can see that the omnivores had higher serum levels of TMAO than did the vegetarians or vegans. TMAO is derived from a number of sources, including animal meat, including carnitine, choline from fish, and glycine betaine from vegetables. It's kind of complex. But if we recode the entire cohort based on their Mediterranean diet compliance, you see that the higher the compliance to the Mediterranean diet, the lower the TMAO values, even though a substantial proportion of these individuals would be categorized as omnivores. So let me make that super clear. If you are an omnivore, you can mitigate your risk for bacterial-linked atherosclerosis by having some degree of adherence to Mediterranean diet. You don't have to go the full hog, no pun intended, and be vegetarian or vegan. So around that time, we became involved in a European Union project, which gave us a truckload of cash to do a transcontinental dietary intervention. This is why people in Ireland love the European Union in contrast to our British neighbors, because it allows us to do stuff like this. So in 5 European countries indicated by these red circles, each of a cohort of 250 people was recruited. Their microbiome was profiled at baseline, and then they went into either the control group or the med diet intervention group for 12 months. And all of the samples were sent to Cork for microbiome analysis. Some descriptive stats, which I can gloss over. Their diet at the baseline was different from Poland, Poland and Netherlands through to UK, France, and Italy, and they had correspondingly different microbiomes at baseline, which you would predict. And if we compare intervention versus control at dietary data level, the intervention did work. I told you I'm not a nutritionist, but even I can understand this. This is the analysis of what they did. I used to think that a med diet was pizza and pasta and Nutella and panna cotta, but no, it's eating boring stuff like whole grains, fruits, vegetables, legumes, fish, reduction in alcohol consumption, reduction in sweets. But I'm sure you all know this. Anyway, the med diet did not alter microbiota alpha diversity. These show the 5 countries pre and post in both control and med diets. And if you look quickly across those, you can see there's no significant difference. So the med diet, either as a treatment effect or comparing between groups, did not improve microbiota alpha diversity, which was a little bit disappointing. But we began to realize that alpha diversity was a poor measure for microbiome quality, because when we adjusted for the baseline microbiota and the level of compliance, and this was critical, in a trial of this scale, you're going to have people whose compliance ranges from very good to very bad. And that means somebody in Poland who had essentially a 0 adherence to a med diet score going to a very high adherence, you could see a microbiome response. And we were able to identify diet positive taxa whose abundance was improved by adherence to the Mediterranean diet, and diet-negative taxa whose abundance decreased in response to the Mediterranean diet. Their names are not important, but if you've been exposed to any microbiology, you will recognize these as the usual suspects of good bugs for dietary trials and for dysbiosis, and bad bugs whose abundance tends to be higher in a variety of dysbiotic conditions. Importantly, when we looked at the correlation between the abundances of the diet-positive taxa and the diet-negative ones, let me deal with the diet-positive taxa first. So these are the desirable ones whose abundance increased on the med diet. Increase in their abundance correlated with reduction in inflammation, gait speed or walking time, and frailty. So all of these health indices improved. In other words, they became less inflamed, less frail, and they were able to walk faster. And at the same time, there was a negative correlation because we had increases in anti-inflammatory markers, cognitive function, and grip strength. And for the diet-negative taxa, the exact opposite observations were made. So this was the first definitive evidence that consumption of a med diet maintained the microbiome and delayed onset of frailty by maintaining the level of these measured health indices, because the people on the control arm had a statistically and clinically significant reduction in those markers. And it includes measures of inflammation like high sensitivity CRP. And the two classical measures for inflammation in the elderly are CRP and IL-6. Now, as I said, like, nutrition was a black box to me, and I am a long distance runner, so I recognize the value of nutrition and I try and eat good stuff, but I'm only human. I became involved in another project which is related to inflammation. It's a bit of a digression because these subjects are not old, but it's kind of cool nonetheless. So this was a collaboration with Max Neudorp in AMC in Amsterdam and his doctoral student Anne-Marie Koopman. And Max was a pioneer who had developed fecal transplantation for type 2 diabetes and metabolic disease. And he published some of the seminal original papers showing that fecal transplant from a lean donor to somebody with metabolic disease improved peripheral and central insulin resistance. And the effects tend to be short-lived, so it's not a done deal and is still actively being researched. So Max and I wondered if the med diet could augment the effect of fecal transplant. So what we did, and the experimental design was a bit flawed, as you'll see in a moment, is we recruited people who 24 males with metabolic syndrome, divided in half, and either gave them their old stool back or gave them an allogenic fecal transplant from a lean donor. And 2 weeks prior to the FMT, we put them on a med diet. And then we maintained them on that med diet for 6 weeks, and we did fecal profiling throughout, and then state-of-the-art uremic clamp to measure their metabolic status at 2 critical points in the experiment. And what we saw was that the med diet alone over the first 2 weeks reduced energy intake. The fat was around the same, saturated fat significantly reduced, carbohydrates were down, fiber was up, and protein was around the same, which is pretty much what we had hoped to achieve. There was a modest change in the microbiome due to the med diet. Now, if we go looking really hard, the summary statistics like diversity and gene counts are hopeless. Beta diversity is not changed, but we can see, and we did report some low-level taxa whose abundance did alter. The cool thing was that between week -2 and 0, which is the lead-in period, we saw already reductions in body weight, which were as good as we were going to get because the subsequent FMT had no effect. So the improvements in body weight, fasting glucose, HOMA-IR insulin resistance, and cholesterol were all achieved by 2 weeks of med diet alone. And the FMT, even from a lean donor, had no improvement upon that. And this is why I think the design was bad. We should have started the med diet later on. So to summarize, and I'm getting to the end of the talk, summarize this part, prebiotics in the elderly had modest effects on microbiome and health markers. The whole diet change was effective, and the med diet eclipsed fecal transplantation and metabolic disease with an inflammatory component. Now, In case you're interested in microbiology, I realize in retrospect that the abstract was probably a little more downbeat in terms of how much we can define a health-related or an unhealthy microbiome. And the big picture at a cartoon level is that we want to promote healthy aging. So we need to delay the onset of these so-called bad bugs or pathobionts. This is an operational name for bacteria which are present in most people, but which seem to outgrow and are associated with lower health. So we want to delay that, and we want to retain a bunch of protective taxa for longer in life, and we want to retard the onset of inflammation and retard the onset of frailty and degeneration. In other words, to promote health span and not just lifespan. And if you are interested in using diet as a lever for doing this, we have now published multiple papers, and this is a recent one in Nature Aging, giving you a short list of bad bugs which positively associate with a new index called Kendall uniqueness, which we've presented in the literature. And then these are more desirable ones which have a negative association with this Kendall uniqueness. So there are shopping lists of bacteria out there if you want to develop food products and want to know which are the good bugs and which are the bad bugs that your food product should be aimed at. Where do we go from here? To summarize at a global level, I contend, and I hope you believe me, that we know what microbes are desirable for healthy aging. And I've provided references for that. We know the med diet is effective, but we don't really know why. Now, practically speaking, I don't know about where you are, but in Ireland, you probably would struggle to eat a med diet year round. You could probably get the legumes and the pulses, but you couldn't get the fresh fruit, not all year round, at least maybe not affordably. So even though you might aspire to switching consumers to a med diet, it's hard for anybody to change their diet, especially an older person, because they become quite habituated. So that's difficult. It worked in the New Age study because we gave them free food every week. You could administer live microbes, and as a proponent of originally probiotics and now next generation therapeutics, I believe in that too. And I've referenced a paper where we cultured the so-called microbial culture collection, 100 organisms, which constitute the most abundant ones. And we're revising a paper at the moment defining a small consortium of 7 microbes for healthy aging. But there are technical challenges to that because you have to produce them anaerobically. Or else you could develop food products that emulate a med diet. So you could say, well, I know that my consumers are not going to change their whole diet. Maybe I can design a new food product which captures the best ingredients. And then the challenge becomes which one, and will they work in isolation and in those consumers? I do want to leave time for questions, so I'm going to wrap it up here. Acknowledge my colleagues, especially a super postdoc who's now gone back to a faculty position in India, Tarini Ghosh, my friend and colleague Fergus Shannon. I'm surrounded by a football team of great colleagues in the APC, core facility scientists, and I've enjoyed generous funding from Irish and European agencies, for which I'm very grateful. And I thank you for your attention.